open up. Okay, anybody have any questions on anything at this point? Before we get started on blood vessels, I want to wrap blood vessels up today and maybe even start the urinary system.
And then after that, there's an exam, right? After urinary
After
system?
blood vessel.
Oh, after blood vessel,
yeah.
so we'll have an exam on. Okay.
Right, right.
Next year.
Yeah, it's I opened it yesterday accidentally, but it's okay. Because we'll finish it today.
So we can take it tomorrow.
You can take it. I think I gave you like a week to take it. So
Okay.
yeah. So anyway, okay. Lab this afternoon, we're going to take a look at the first half of the digestive system. That's why we have the digestive system model on the screen there. That was the camera that came up. So it's a long section. So I'm going to divide it into parts this week and next week. Otherwise, we'd be here till 3:30 with me talking about movement of food through the digestive system, which is not as exciting as it sounds.
It doesn't sound very.
Okay, so said the woman with the unknown baby wipe.
I know, honestly.
Yeah, so, okay. So I'm going to go ahead and move us into the PowerPoints. And I don't know if this is exactly where I started, but this is where I'm starting. I mean, where I stopped. This is where I'm starting today, because this is a good place to talk about how we control our blood pressure short-term and long-term. So let's go ahead and bring it up. And hopefully your slides mostly match mine. I noticed when I was going through the slides the other day, when I looked into the second half of it, I said there was a whole complete extra set of slides that didn't belong there. So of the same stuff I'd already said. So if you see extra slides, the ones I didn't add anything new. I just cut stuff out. So, okay. Short-term and long-term controls of blood pressure. Our blood pressure fluctuates all the time. You walk up a flight of stairs. You drive through busy traffic. You deal with stresses all the time. We're going to always see our blood pressure go up and down. You know, never rush when you're going to a doctor's appointment. Try not to rush when you get there, because if you're rushed and stressed, your blood pressure will be up, and have to take it again after you relax a little bit. So, but we always move. Our blood pressure is always changing for us. It is the long-term control or the long-term issue that gives us health concerns. So short-term, we can open and shut blood vessels. Open and shut blood vessels. We can regulate where the blood's going. We can it says altering peripheral resistance. The best way to alter resistance in our blood flow is through vasoconstriction or vasodilation of the arterioles. Long-term control deals with blood volume, like how much water do we take in? How much water do we retain? And this gets into the whole issue of eating salt or avoiding too much salt, because if you ingest salt, you're going to take in water. That's just the nature of water. You know, the old saying, water follows salt, which is true because of osmosis. If you eat a lot of salt, you're going to make your plasma salty, and water is going to move into your plasma through osmosis, because you have, if the plasma is salty, it's going to have less water. Salt concentration is going to be greater than the interstitial fluid. Water will move into the plasma. Your plasma levels will increase. So you eat salt, you'll increase your blood volume. If you increase your blood volume, your heart has to work harder to pump that blood. So we have long-term mechanisms to control our blood pressure, deal with blood volume issues. So now for peripheral resistance, I know we talked about this the other day, because I know we talked about baroreceptors. Peripheral resistance, we move blood around by opening and shutting vasoconstricting and vasodilating the arterioles. And it's all very, very rapid. We move it around. It's the same mechanism we use to get rid of body heat on a hot day, or to retain body heat on a cold day by altering the movement of the blood into the capillaries. So, we have demands. We're hot, let's get rid of the heat. We're cold, let's retain the heat. But we also monitor things like pressure. We can monitor pressure. We have the baroreceptors that are embedded in our carotid sinus and in our aorta, in the aortic arch. We have baroreceptors there that monitor the pressure. So a short-term rise in pressure, we can bring that back down by causing vasodilation of arterioles somewhere else. It brings the pressure down because these baroreceptors are reporting right to the medulla oblongata. And the last bump in the brainstem is the medulla. The medulla is full of reflex centers. It's also where we have the cardiac acceleratory center and inhibitory center. It's also where we have our respiratory centers. All this stuff is in there. The vasomotor centers that control the vasoconstriction and vasodilation are in there. So if our pressure is detected to be elevated, short-term elevation, the pressure in the aorta, the pressure in the carotid sinus, we can fix that by how we dilate or constrict the arterioles. And that involves the smooth muscle, because the vasomotor centers will stimulate smooth muscle to constrict or relax. Now, the vasomotor centers use norepinephrine. Norepinephrine gets released. Norepinephrine is a neurotransmitter. We all remember neurotransmitters, right? Just, okay. Yes, thank you. So, thank you. Yes. Neurotransmitters can either be excitatory or inhibitory. Norepinephrine is excitatory in smooth muscle. It causes it to constrict. The more norepinephrine you release, the more the smooth muscle around the arteriole will constrict. It will reduce the flow. It will increase resistance. That can raise pressure. On the other hand, if we want to lower pressure, all we have to do is stop releasing the norepinephrine. And so when we do that, the smooth muscle relaxes, the vessel dilates, and so our pressure drops. So this is a short-term fix. You run up a flight of stairs, I don't know why, but you run up a flight of stairs and your blood pressure goes up because your heart has greater demands. Your heart's demanding more oxygen and more. It wants to get rid of CO2 because you run up a flight of stairs, you burn glucose. So you got to get rid of the CO2 and you got to get some oxygen in there. Probably not, because it wouldn't be, in 10 seconds, you wouldn't be using any oxygen, but your heart's still going to beat faster to get rid of any waste products or get more glucose. It's probably the key is glucose. Your heart's beating faster. When your heart beats faster, your blood pressure goes up. So we bring it back down because we detect that change in blood pressure. We're not even aware of it. Now, the vasomotor center and the cardiovascular center in the medulla are going to fix all this for us. These are all, the vasomotor center is a sympathetic nervous system structure. The vasomotor center will release norepinephrine. This is where the action happens. The vasomotor center will tell the arterial smooth muscles to constrict by sending out norepinephrine, or they'll tell it to relax by not sending out norepinephrine. It's all about the norepinephrine. You either send out a lot of it for constriction, or you don't send out very much at all. It isn't like we send out norepinephrine to constrict and epinephrine to relax. We just send out a lot of norepinephrine, or we send out a little norepinephrine. And so we innervate, as they say, the smooth muscles, because it's the smooth muscles that are constricting. They're constricting or relaxing. Now, that's the vasomotor center. The cardiovascular center also includes the mouthful there, the cardiac acceleratory center and the cardiac inhibitory centers, which control the vagus nerve and the cardiac nerve. Vagus nerve sends epinephrine vagus nerve sends acetylcholine to the SA node, the AV node, and slows down the heart. Vagal tone. If without the vagus nerve there, our resting heart rate would be around 100, but our resting heart rate's about 70 because we keep dumping acetylcholine into the SA node, slows our heart rate down to what we consider normal. We want to speed up our heart rate. We send epinephrine on the cardiac nerve from the cardiac acceleratory center to the SA node, the AV node, and the muscles of the ventricle. Heart rate goes up. And we know that when the heart rate goes up, our blood pressure goes up. When we know that when the heart rate goes down, our blood pressure goes down. The faster the heart has to beat, the greater the pressure is going to be. But this is all short term. If your heart rate goes up, your blood pressure goes up. We could even demonstrate that here if we had somebody stepping up and down. If we had to set a step stool here and you just stepped up and down for 20 times, one after another. If we took your blood pressure before that and after that, and took your pulse rate, your heart rate would be going up, and your blood pressure would go up because your heart's beating faster. It's pumping more blood. It's putting more blood into the arteries. And it's going to give more resistance against the arterial wall, and you're going to have higher pressure. That's all short term. I think I've said all this already. Sympathetic nervous system is going to be the driver here for us. If you remember from AP1, the sympathetic nervous system only uses epinephrine and norepinephrine as its neurotransmitters. The parasympathetic system uses acetylcholine. So we only got 3 neurotransmitters here we're playing with, and sympathetic nervous system has 2 of them, epinephrine or norepinephrine. And so all we're doing to either raise or lower blood pressure here is simply releasing norepinephrine or releasing epinephrine into the heart, or not. Because if we don't release it, everything relaxes. We have less. If we don't release norepinephrine, we have less constriction. We have dilation of the smooth muscle. It relaxes. If we don't release epinephrine into the heart doesn't beat as fast. The parasympathetic side is dominant. We keep releasing acetylcholine in here. And we depend on the baroreceptors, the chemoreceptors, any hormones we might have in our system, any chemicals floating around in there, like caffeine, for example, or nicotine, anything that functions as has some sort of excitatory effect. And we don't really monitor oxygen that much. We really monitor CO2. We monitor CO2 indirectly through pH of the blood, because our medulla makes the assumption, sometimes incorrectly, that if our blood pressure, if our pH drops and we're becoming more acidic, it's because we're generating more CO2, which is normally the case. You exercise, you burn up glucose, you release carbon dioxide from the Krebs cycle. Oh, I said a bad word, Krebs cycle. But that's normally the case. But what happens? Remember what we talked about in endocrine ketoacidosis. What does ketoacidosis do? It increases the presence of acids in our blood as we break down fats to try and convert them to glucose. People with ketoacidosis have a very low blood pH. The medulla doesn't know it's not from glucose, not from exercise. All they know is that the pH is low. Okay, well, let's breathe faster then when our blood pH is low. That's, it's like the #1 answer for the medulla to say, well, if our pH is low, let's breathe faster. Most of the time it's correct. We only monitor CO2 indirectly, but that normally covers oxygen levels and covers hydrogen levels, hydrogen ion levels and stuff like that. Okay, now, it works both ways to regulate our blood pressure in the short term. If our pressure goes up, we respond by allowing the smooth muscle to relax around the arterioles, our vessels enlarge, our blood pressure drops, the resistance drops, the heart rate slows down, and the cardiac output slows down. All this goes down because we say, hey, our pressure's up, let's bring it back down. So we stop releasing the norepinephrine. If our pressure drops for whatever reason, then we start going the opposite. We start releasing epinephrine to increase the heart rate. So we increase the cardiac output. We vasoconstrict the arterioles. We raise the resistance. Every time we increase resistance, we raise blood pressure. Every time we increase the heart rate, we increase blood pressure. So it works both ways here. We can either bring our blood pressure down to our normal level, or we can bring it up to our normal level. Now, in addition to the pressure receptors, we have chemoreceptors that can detect changes in carbon dioxide levels. We have these chemoreceptors located right next to the baroreceptors. So we can monitor changes in CO2 and indirectly oxygen levels too. And so right in the same location in the aortic arch and in the carotid sinus, we are going to monitor for changes in CO2, indirectly oxygen and indirectly we detect it through pH more than anything. And then we can regulate our pressure depending on what the levels of carbon dioxide are telling us. If carbon dioxide levels are rising in our blood, it could very well be an indicator that our pressure is low because we're not pumping as much blood. We're retaining more blood, and if we're not sending more blood to the lungs to get rid of the CO2. So these chemoreceptors are also a way to detect this. But this is, again, all part of our short-term mechanisms. Okay, so what about hormones? What do hormones do for us? Well, we know that when we get stressed, the adrenal medulla secretes epinephrine and norepinephrine. When we get stressed, you're driving and you're in a hurry to get somewhere. We've all been there. You're in a hurry. And of course, every light is against you, and you've got a million people on the road. It seems like a million people on the road in front of you. Or maybe like this weekend, you're trying to go somewhere with the rod run people in the way, And of course, they all have to stop and look at the same 37 Ford they saw 10 feet earlier. And I'm not picking on rod run cars. I love cars.
It's terrible though.
But oh no, I have said very unpleasant words about traffic, and I've been stuck in traffic. So, and we've all experienced that. But that's neither here nor there. But that's the kind of stress, because you won't see me anywhere near Pigeon Forge after today.
Yeah.
So.
You sound like my papa. He said, I ain't leaving the house after tomorrow.
Oh yeah, well, see, yeah, that's.
He lives near Old Mill, so.
Oh, that makes more sense. Yeah, I live here in Sevierville.
Yeah.
And I won't go anywhere. I won't go anywhere past. I won't even go to Walmart.
No, you can't go.
You can't.
Make it.
Just go to Little One.
Oh, I know.
I like that.
Actually, I get mine delivered. It's even easier.
Oh, fancy.
Yeah, it doesn't cost any more. But what I save in having to drive to the Walmart and deal with
the stress.
And the stress of Walmart. Yeah. So I probably won't go past.
I have to work all weekend.
I'm sorry.
No, it's okay.
It's okay. So
Not really.
I know. So anyway, now that we've established that there's a lot of stress around here this weekend, when we get stressed, we release epinephrine and norepinephrine. We raise our heart rate. We vasoconstrict our vessels. We're sitting in our cars and nobody's moving, and we're very annoyed. Our blood pressure goes up. Our heart rate goes up. That is the actions of the hormones, raising our heart rate up. However, we do have several mechanisms here to try and counteract that. ANP, atrial natriuretic peptide, which is why we call it ANP. When the atria stretch, when more blood, because your heart rate's up, you're vasoconstricting, so your heart's working harder, and there's more blood coming back into the atria. They stretch. When they stretch, they act on the kidneys. ANP acts on the kidneys to allow us to excrete more sodium. We pee away more sodium. Now, how does that help? Well, where's that sodium coming from? Everything we eat, everything we drink ends up in our digestive system. The sodium was in our plasma. Whether it got filtered through our kidneys or not, it will eventually end up in our kidneys. All of our plasma, our blood's filtered through our kidneys 60 times a day. So all that sodium that's in our plasma is going through there. If our pressure is up and we get rid of sodium, the water's going to follow it. Just like water runs into the blood if we eat a lot of salt. If we get rid of a lot of salt, water's going to follow it. So we excrete sodium, and that lowers our blood volume. Getting rid of sodium affects the presence of aldosterone. Aldosterone tells us to normally retain sodium and raise our pressure, but ANP can work against aldosterone. By getting rid of the sodium, it counteracts the effects of the aldosterone and the renin, and so we vasodilate, we lower our blood pressure, we lower our blood volume. So our heart itself can try and fix the problem. The atria, when the atria get stretched, they release ANP. It's a short-term fix. Anti-diuretic hormone, our old friend from telling us when to pee and not to pee. Anti-diuretic hormone, if our blood pressure drops, we will release anti-diuretic hormone. Something I didn't tell you about anti-diuretic hormone at the beginning was it not only does it restrict, anti-diuretic, the anti-pee hormone, not only does it retain water in your system by not letting you pee as often as you did before, but it also functions as a vasoconstrictor. We call it vasopressin. Here, oh, we got a comment. Yes, ANP counteracts the stress of ANP. Possibly. If ANP is stressing you, then ANP is definitely going to work against that. So, okay. It does sound funny. Yes, it does. So,
Who said that?
Logan said that.
Good job.
Yes.
Great.
So, okay, let me make a mark here on the grade book about Logan. So, you wanted stress, I'll give you stress. So, okay. Yeah, I know. Okay. Now, anti-diuretic hormone works as our friend. The reason we don't pee is to retain water. If we retain water, our blood pressure goes up. Sometimes that's a good thing. When our blood pressure is low, we can inhibit anti-diuretic hormone, and we pee more often, and we'll also vasoconstrict. So anti-diuretic ADH works both ways. Blood pressure is low, so we don't pee and we vasoconstrict. Because if we don't pee, we retain that water. If we retain that water, our blood volume goes up and our pressure goes up. If our pressure is high, we release anti-diuretic hormone, we pee, and we relax the vessels. Because it's either we release the anti-ADH and we don't pee and increase our volume, or we don't release it, we don't vasoconstrict, and we don't raise our volume. So ADH can go both ways on this. Now, angiotensin 2. Angiotensin 2 is released when our blood pressure is low. Our kidneys detect low blood pressure, because remember, we filter our entire blood supply through our kidneys 60 times a day. So our kidneys are very, very aware of our blood pressure. And if our blood pressure is low, the kidneys will release renin. And renin stimulates the conversion of angiotensinogen to angiotensin 1 to angiotensin 2. Now, angiotensin 2 could just stay there, or it can trigger the release. It also can trigger the release of aldosterone. Angiotensin 2 is a vasoconstrictor. So if our blood pressure is low, the kidneys detect that, release renin, convert angiotensinogen into angiotensin 2, causes vasoconstriction, the pressure goes up. Anytime we constrict, our pressure is going to go up. Our blood pressure is low, the kidneys detect it, and they release angiotensin 2. Now, that's short term. Long term, yes, I agree. We all are too. So long term, angiotensin 2 triggers the release of aldosterone. Aldosterone is the number one mineralocorticoid from the zone of glomerulosa. Remember that back in January, we talked about the zone of glomerulosa. It's the outer covering of the cortex, it's the outer layer of the cortex. It's where all the mineralocorticoids come out. Aldosterone is the number one mineralocorticoid. It regulates fluid levels and sodium levels in our blood. Aldosterone tells us to retain sodium. Sodium causes us to retain water. When we retain sodium and water, our blood volume goes up and our blood pressure goes up. So aldosterone, this is a long-term fix. If our blood pressure is too low, we will retain salt and retain water. So we see the role of angiotensin 2 here from the kidneys. The kidneys play a real important role for us in blood pressure. And so they're always monitored, since they're monitoring blood pressure constantly, as all the blood plasma has to go through the kidneys constantly, 60 times a day. So they're very much aware of the pressure. When we get into the kidneys, we'll see that pressure is important on how we filter our blood. If the pressure is too high, we filter too rapidly and we miss stuff. If it's too low, we filter too slowly, and waste products have time to get back into the plasma. So our kidneys are always monitoring blood pressure. Short term, we can stimulate, cause vasoconstriction to raise the pressure, or not and let it drop. Long term, we trigger this whole renin, angiotensin, aldosterone. Aldosterone, again, I can't emphasize enough here. We retain sodium. And we retain water. Yes, to answer Logan, to answer your question, problems with our hormones can affect the heart rate and blood pressure. If we release aldosterone, we raise our blood pressure because we're going to raise our blood volume. If we don't release aldosterone, we can lower our blood pressure and lower our blood volume. We can control the heart rate with these hormones. I mean, ANP is a hormone. And I made this point a long time ago, well, not that long ago, but about different types of hypertensive medications. If you interfere with the sequence of making aldosterone or making angiotensin 2, you can control blood pressure. If you because two of the most useful blood pressure medications work by interfering with the angiotensin sequence, whether they're an angiotensin converting enzyme or an angiotensin angiotensin blocker. And they're called ACEs and ARBs. The beauty of these is you don't produce angiotensin 2, so you don't get vasoconstriction and your blood pressure stays low, and you don't get aldosterone. So there is a great deal of impact on hormones on our blood pressure and our heart rate. So now, let me get back down here. Great question there. Now, this is why we don't like to see our patients with hypertension eat a lot of salt. Because if you eat a lot of salt, what are you going to do? You're going to retain water. Because when you eat a little bit, even a little bit of salt changes the salt concentration in our plasma, which makes us thirsty. And so when our plasma salt concentration goes up even a little bit, we start drinking water, other fluids, it doesn't matter. We're drinking something. Why do you think a restaurant that serves chips and salsa to you as your appetizer, nice, hot, salty chips, why do you think they give us the salty chips?
Just to get you to buy more drinks.
Exactly, to get us to buy more drinks. Because drinks are a huge profit center for restaurants. Because the markup on drinks is like, 3 million% or something. It's like a Coke costs them a penny and they charge $3 for it, or even a glass of tea, which costs them what, 1/10 of a cent to make a glass of tea. They make, they're charging $3 or $4 for a glass of tea. Not even more profit on an alcoholic beverage, which in its own way doesn't do anything for us anyway, but it does for them because, alcohol inhibits anti-diuretic hormone. So you drink enough alcohol, you start having to pee, you pee enough. What do? You get thirsty and you drink more beverages. So not that there's anything wrong with any of that. So I like my chips and salsa as much as anybody does. So, okay. Now, I already mentioned some of this. Nicotine is a vasoconstrictor. A person, among other things, nicotine releases dopamine in the pleasure centers in the brain, but it also causes vasoconstriction, so the blood pressure goes up. So that dopamine that's getting released goes around the systemic circulation faster. So you get an overall sense of well-being. A smoker has that sense of well-being on their first puff of their first cigarette of the day. The downside is that every cigarette after that can't trigger as much of a dopamine release as it did before. But the smoker will continue to smoke because they keep wanting to get that buzz from the dopamine. It's not a buzz, but it's a sense of well-being. Each cigarette after that gives a little bit less, a little bit less, a little bit less, but the smoker will continue to smoke to try and recover that effect from the first cigarette. That's what makes nicotine addictive. It's because of this dopamine release. So it's a vasoconstrictor. You feel good from the dopamine and it raises your blood pressure. You feel more alert and awake there on that cigarette. Nitric oxide is a vasodilator. It's a systemic vasodilator. We've talked about that in the heart. Person with angina. Angina is chest pain caused by reduced oxygen delivery to the myocardium. So it's called ischemia. To stop and think of the word again. It's called ischemia. So you're not getting enough oxygen delivery to the muscles of the heart, and it causes temporary chest pain. And your patient will take a nitroglycerin tablet and it will get a systemic vasodilation. It's not just the heart, it's the whole body. Histamine is a vasodilator. Why does your nose run when you're exposed to pollen? Because pollen lands on the histamine receptors in the nasal cavity, and the reaction is to have vasodilation of the blood vessels in the nasal cavity. And maybe not only do they vasodilate, but the capillaries become extremely leaky and plasma leaks out as mucus. And our nose runs. It leaks out into the mucus layer. We have more mucus there. Our nose runs because of that. And the reason it does is to try and flush away the irritant. That's what histamine's doing for us. Our eyes water for the same reason. Now that is usually the only reaction, you know, if we're allergic to pollen of some sort, that's usually the reaction we get. Now, if you're allergic to seafood or peanut butter or bee stings, you know, and any one of those are also going to trigger the release of histamine. Now, histamine becomes a real health concern if it causes if it does cause a systemic effect. You're allergic to bee stings. You're stung by 3 bees and you swell up to about 4 times your normal size because your blood vessels have vasodilated and they have become very porous, leaky to the plasma, and all your plasma is moving out into your tissues. That's where you're getting the edema. You're swelling because of systemic edema. That water that's going into your cells and in your tissues, it's coming from your plasma. What's happening to your blood volume? It's crashing. So a person who is allergic to a bee sting has the risk of asphyxiating because they swell, their airway swells shut because all the soft tissue is pushing around it, or they die because blood volume has dropped below the point where it can carry blood. So that's why you use a person that's allergic to things like bee stings carries an epinephrine pen, an EpiPen, to inject themselves with epinephrine to cause increased heart rate, to cause vasoconstriction, and not die, essentially. So normally histamine is just an annoyance. If you're allergic to pollen, for example, or cat hair or dog hair or something like that, your nose runs and your eyes water and you sneeze, histamine's doing its job. We take antihistamines to block that action when we're fighting, when we're dealing with pollen, dealing with allergies, things like that. Alcohol is another chemical that interferes with our blood pressure. Alcohol causes our blood pressure to drop. Given enough alcohol, it will kill your patient. So if they ingest enough of it, because it will depress the vasomotor center. It will also allow us to lose fluid because alcohol inhibits anti-diuretic hormone. So we pee more frequently. But by blocking the actions, by inhibiting the release of ADH, there's nothing there to tell us not to pee. So we pee and we pee and we pee. And our volume drops. It also causes us to vasodilate. So we're losing blood volume and we're vasodilating because it depresses the actions of the vasomotor center in the medulla. And so your patient, given enough alcohol, they will shut down through alcohol poisoning. Now, what about long-term? The kidneys are involved with long-term regulation. I alluded to this a little while ago when I talked about angiotensin and aldosterone. The kidneys can alter our blood volume very easily for us. Remember, they filter our blood 60 times a day. They filter out all these waste products for us. If the blood pressure changes, the kidneys will respond to it. See, the pressure receptors in the carotid sinus and the aortic arch can adapt. They can get used to the higher pressure. Why do you think our blood pressure, if a person is starting to develop hypertension at age 30, if it's uncontrolled, why does it just continue to increase year by year? the baroreceptors that would normally be sending a signal to the vasomotor center saying, hey, you know, our blood pressure is getting high, we need to do something about this. They just get used to it. Our bodies get used to stress. We don't like it, but our bodies get used to stress. And so the baroreceptors adapt to pressure changes. The elevated pressure is now the new normal. So, but the kidneys don't. The kidneys can work to fix blood pressure. The kidneys will not adapt. The kidney, if the pressure goes up, if our blood pressure goes up, the kidneys will try to fix that by getting rid of more water. Less water means less volume. That water comes out of the plasma. So you get rid of more water. So if the pressure goes up, our kidneys will tell us to pee more frequently to get the water out of the system. If our pressure goes down, the kidneys will retain water. So it's a long-term fix there that the kidneys are trying to institute. Okay. Yes, the upregulation and downregulation. Same type of approach here with the hormones. You know, we can, you know, because what we're doing here is ultimately affecting aldosterone. Yeah, upregulation when we adapt to the higher pressures, and downregulation when we adapt to the lower pressures. So good point. So, okay, now. There's two ways the kidneys are going to affect our blood pressure. The first one is to change blood volume. If blood volume drops, pressure drops. It's as simple as that. If there's less fluid in the blood, our pressure drops. If we retain fluid, then our pressure goes up. That's the one way. That's the direct mechanism that the kidneys use to alter our blood pressure. Get rid of the fluid or keep the fluid. Raise the blood volume, our pressure goes up. Drops the volume, our pressure goes down. Indirectly, we come back to our friend renin-angiotensin. Back to our friend renin-angiotensin. And I know it sounds like I'm, you know, repeating myself, and I am, because this is the knowledge about renin-angiotensin. There are so many things we use this for. So if our blood pressure drops, the kidneys will release renin. I said this already, but I want to say it again because it's a really busy slide, but it's important. If our blood pressure drops, renin will get released from the kidneys, which triggers the conversion of angiotensinogen, a protein, plasma protein, to angiotensin 1 to angiotensin 2. Angiotensin 2 does two things. It's a vasoconstrictor to raise blood pressure. It also triggers a release of aldosterone from the adrenal cortex. So if we look here at our flow chart, here we have the decreased arterial pressure. The kidneys are going to release renin. So our blood pressure has dropped. The kidneys release renin. Renin triggers the conversion of angiotensinogen ultimately to angiotensin 2. Angiotensin 2 is a vasoconstrictor, which raises blood pressure. Bypass all this other flow trigger. We make angiotensin 2, it functions as a vasoconstrictor, pressure goes up. Angiotensin 2 also triggers the release of aldosterone from the adrenal cortex. Aldosterone tells us to retain sodium. When we retain sodium, we retain water. That increases blood volume. When we increase blood volume, we increase pressure. So even if it's the pressure receptors telling us what's going on, they still end up telling the kidneys, hey, by the way, we need to get some aldosterone made. So the key here to raise our blood pressure is for the kidneys to either retain more water or to release aldosterone to tell us to make more water. So now if you're trying to control blood pressure, you probably want to block the action of the angiotensin 2. Angiotensin 2 is a vasoconstrictor. It also tells us, it also releases aldosterone. You got two different things going on here that are going to increase blood volume, increasing blood volume by retaining sodium, which causes us to retain water, or you are going to vasoconstrict. Two different mechanisms in place here with angiotensin 2. Now, if we can control angiotensin 2, we can keep blood pressure from going up. And many of our blood pressure medications are either what we call an ACE inhibitor, an angiotensin converting enzyme inhibitor. It prevents the conversion of angiotensin 1 to angiotensin 2. So if you're trying to control your patient's blood pressure, you put them on an ACE inhibitor. They don't get a chance to make angiotensin 2, which means they don't have vasoconstriction and they don't make aldosterone. You control blood volume and you control vasoconstriction. The other medication is called an ARB, angiotensin releasing blocker. It blocks the release of angiotensin 2 from the kidney. Or we're not, essentially we're not converting angiotensinogen to angiotensin 1 to angiotensin 2, so we don't get the vasoconstrictor. We don't get aldosterone. Both of these work pretty much the same way. One interferes with the ACE inhibitor, which is the more common of the hypertensive medications, works by preventing angiotensin 2 from forming. It blocks at the level of angiotensin 1 to angiotensin 2. The ARB, the angiotensin releasing blocker, prevents the formation of angiotensin 2. In both cases, you don't get vasoconstriction, you don't get sodium and water retention. And why are there two different ones? Because ACE inhibitors, the more common of the two, in some percentage of the population causes a dry hacking cough. ARBs were invented because they do essentially the same thing without the nasty cough. So about 30% of the population that take ACEs are going to have that cough, and they're on ARBs. That's the only difference in here. So if you have a patient that's on blood pressure medication, they complain about a cough all the time, a constant cough, a constant dry, irritating cough, as we say, a non-productive cough, and they're taking an ACE inhibitor, it might be worthwhile to look into an ARB. And there's all sorts of medications. There are all sorts of ACEs and ARBs out there. Okay. That's why we pay attention to the kidneys in long-term blood pressure control. Because remember, if volume goes up, pressure goes up. If volume goes down, pressure goes down. If heart rate goes up, pressure goes up. If heart rate goes down, pressure goes down. Okay, now how do we know if the circulation is efficient? Well, this is where we take the vital signs. Something as simple as blood pressure, pulse, respiration, and body temperature. Just what are collectively called the vital signs. Blood pressure, we take the systemic pressure at the brachial artery. We place a blood pressure cuff, a sphygmomanometer, on the brachial artery. We inflate the cuff until we no longer hear any sounds through a stethoscope in here. We pump it up to what we think is a reasonably high enough number to block the brachial artery. We hear no noises. We hear no pumping sounds in here. Then we release the pressure, and then the first sound that we hear is the systolic pressure. Whatever value on a pressure gauge tells us when we first hear the sound, that's the systolic pressure, and we release the pressure. And when the sound goes away, that's the diastolic pressure. So we get our systolic and diastolic pressure right there. The pressure at the point of contraction, systole, and the pressure at the point of relaxation, diastole. That's one of the vital signs. Also, pulse. Pulse is the pressure wave, the expansion and rebounding of the arterial wall as a new surge of blood comes out from the heart every 0.7 of a second. That surge of blood under pressure shoots out, the artery wall expands and then contracts. And it's that snapping back of the artery wall that gives us the pulse. We usually take it at the radial pulse, right here behind the thumb, right at the radial artery. Our pulse is a good indicator of activity. It's a good indicator of health levels. Is your patient, someone who exercises and is in good physical condition? Do they have underlying health issues? Are they not as fit as they could be? The pulse gives you that, it can tell you right there. Are they stressed? Did they just drive through run traffic to come to the doctor's office because they're always complaining about being stressed? That's one where you might want to let them sit for a little while. So, very simple mechanism. Heart, breathing rate, 15 times a minute, respiratory rate, body temperature should be right around 98.6, but it doesn't have to be. Many people run a little bit low, 98.6, 98, 97.8, somewhere in, it's whatever is normal for them is their normal. Some people are always going to, very rarely do people run right at 98.6 or 37 degrees C. That's not always going to be what you see. When you have your blood pressure taken, who nails 120/80 every time? Our blood pressure is 117/67 or 118/75 or something like that. We're always, we're in the range. It's very unusual to hit it right on the 120/80. That seems like the magic number. So our blood pressure is not going to be constant all the time. It's going to vary day and night. It's going to be highest in the morning and lowest at night, obviously when we're sleeping. We are going to have our blood pressure change. Generally, we see people, their blood pressure has a gradual increase over time as they age. The weight gain will cause blood pressure to go up because you have more blood volume. Individuals that are overweight or significantly overweight are going to tend to have higher blood pressure. There may be differences in blood pressure between males and females. There may be differences in race. There may be differences in how they feel at the time or how they're standing or sitting. If you've ever sat there, if you cross your legs, they'll tell you to uncross your legs before they take your blood pressure, because that alters the effect. The socioeconomic status. Now, how would your bank account affect your blood pressure? Stress. Yes, thank you. It works both ways, whether you have very little in the bank and you're worried about meeting your bills, or if you have a lot in the bank and you're worried about losing it. A large bank account is not always a good, doesn't always make people feel good. I'll just say it that way. So also physical activity. Is your patient, are they a runner? Do they play sports? Do they engage in physical, different things? Or are they just they just sit around? What do they do? All these things, these are outside influences on our blood pressure. So. Beauty of our body is that we auto-regulate all the time. We meet the demands of the different parts of our body when we need them. We send more blood to our digestive system after after a meal, so we can absorb more nutrients, so we can collect the nutrients that are being absorbed in the small intestine. During physical activity, we send more blood to our muscles. We are going to adjust our organs, adjust the flow to the organs based on the demands. So it isn't like we're sending the same amount of blood everywhere throughout our bodies. We're always going to be meeting the needs. We open and close arterioles as necessary. It gets back to the old example of a hot day and a cold day. When you go outside on a hot day, you send more blood to your skin to get rid of body heat. On a cold day, you reduce the flow to your skin to retain body heat. Autoregulation. We don't have to stop and think about it. The pressure, the mean arterial pressure, is always going to be constant, but we're going to alter the flow. The overall mean arterial pressure is always going to be constant, but we will alter the flow to areas that need it. So what drives autoregulation? Well, if the tissues don't get enough oxygen, they don't get enough glucose, then autoregulation kicks in. We can, a very simple mechanism to cause vasodilation is the release of nitric oxide. Nitric oxide is released from the membrane, the basement membrane of the blood vessel. So we vasodilate. We increase the flow to, this is a short term, this is autoregulation. We don't have to think about vasodilating a vessel to increase the flow to a region that might need more oxygen. So there's, any number of substances that are going to drive this, from a metabolic perspective. If we discover that our glucose levels are low in the tissues, if the oxygen levels are low in the tissues, we will autoregulate. And it usually depends on hemoglobin levels and nitric oxide levels to do that. Yeah. There's a term here called myogenic controls. Myogenic controls. A myogenic response means that we are stimulating the smooth muscle around the vessels. And we can vasoconstrict or we can vasodilate. If we have increased flow, if we have increased pressure in the vessel, we will vasoconstrict. If we're not stretching the vessel very much, we will vasodilate. The smooth muscle around the vessels responds because we don't necessarily, if some is good, more is not better. If we have pressure in the vessel, meaning we have too much blood going to one location, we can vasoconstrict to reduce that. If we don't have enough blood going to an area, we can vasodilate to increase flow to the tissue. If some is good, more is not better. Just because we got lots of blood going to an organ doesn't mean that's necessarily a good thing. If we reduce the flow to that organ, that may be what we want to do. Or if we're not sending enough blood to the organ, we can dilate and send more blood. So this is a myogenic control. This is the response of the muscles This is the response of the muscles, the smooth muscles around the arteries and arterioles. So now, everything I mentioned is all short-term. It's short-term autoregulation. Do we have enough sodium? Do we not have enough sodium? Is the blood flow too high or too low going to the spleen, for example? We can fix that. But what if that isn't good enough? Then we go into long-term autoregulation. Angiogenesis, the creation of new blood vessels. We do this in a number of areas. Angiogenesis, a person who experiences significant weight gain over a period of time will increase the number of blood vessels they have to keep the new tissues fed. So a person who is 100 pounds overweight, 50 pounds overweight, whatever, is going to, or even 10 pounds overweight, is going to create new vessels through angiogenesis to keep those tissues supplied. And if you have new vessels, you have to have more blood. More blood means more blood volume, which means higher blood pressure. So this, to meet the needs of the tissues. See, we got two things going on here. We have trying to control blood pressure, but we're also trying to feed the tissues. The cells need to be supplied, and they're going to work against each other a lot of times, because a person who is overweight is going to have more blood vessels and they're to supply more blood to the tissues to feed them, but they're going to raise their blood pressure. So angiogenesis increases the number of blood vessels to a region. Now we can, we use this in the heart. If the heart has partial blockages, with a patient's heart has coronary artery disease, angiogenesis occurs, so we can get workarounds around the partial blockages, and that keeps the tissue downstream of the blockage alive and functioning. It just doesn't work very well under times of stress. We also develop additional blood flow if we were to live long-term in Colorado, at 10,000 feet, for example. We have half the oxygen available in the air that we have here. So one way to compensate for that is to increase the number of vessels to deliver more blood to the region. You have less oxygen, so you have more red cells and you have more blood. And so that's not a bad thing there. And this is the thing I've been talking about here, about telling us to send more blood to the skin or retain blood away from the skin. When we sweat, it also causes vasodilation. Sweating triggers vasodilation. But as the temperature drops, we send blood with all of its heat deep into the viscera. Now, I have to talk about the lungs just a little bit here. The lungs are different than any other location, and they're very short pathways. The arteries and arterioles act more like veins. The pressure in the lungs is very, very low. The arterial pressure going to the lungs is about 24. The systolic is 24. And diastolic is 8, so it's very, very low. Because remember, we're talking about tissues that are one cell layer thick. Anything greater than that would just blow through them and destroy the alveoli. If we don't have enough oxygen, and this is where we are, this sounds contradictory. In the lungs, when oxygen level drops, we vasoconstrict. When we have high levels of oxygen, we vasodilate. It is the opposite of all the other tissues in the body. Oxygen levels cause the blood supply in the lungs to vasoconstrict, but it raises the pressure in there so that we we have less oxygen, but we're trying to push it through faster. High levels, we vasodilate. Because again, if some is good, more is not necessarily better. Too much oxygen is also not a great thing. So, and we've mentioned this already. The heart with its coronary circulation, the point that's important here is when the heart contracts, the blood flow within the heart stops momentarily. And then when the heart goes into diastole, then the circulation continues in the heart, delivering oxygen and nutrients to the muscle cells. So. When the heart's at rest, it is controlled myogenically, but under exercise, our vessels will dilate in the heart because of a rise in carbon dioxide, because we're burning up glucose. And we can increase the blood flow fourfold. And we keep this constant flow through the heart. Okay, so what happens at the level of the capillaries? I haven't really mentioned this yet. Remember, the capillaries are only one cell layer thick. And we have red cells carrying lots of oxygen. The oxygen carried by the red cells, along with carbon dioxide, we pick up oxygen from the lungs, and we give off carbon dioxide. The red cells are passing through the capillaries. The oxygen can diffuse from the red cell through the membrane of the red cell, through the membrane of the capillary, and then through the membrane of the tissue it's going into. And the waste products can do carbon dioxide can do the same thing. And we deliver the gaseous products to the tissues and or to the lungs. We deliver CO2 to the lungs, and it diffuses out of the lungs. There's a layer of mucus surrounding the inside of the alveoli, and the gases diffuse out of that or diffuse into that. And then the other waste products are carried to the kidneys to be filtered out. So nutrients, glucose, can diffuse across the membrane. Glucose is carried in the plasma, and it diffuses out into the tissues as needed. There's always a greater concentration of glucose and oxygen in the blood than in the cells, in the tissues. There's always greater concentrations of CO2 and waste products in the tissues than in the blood. So it always diffuses passively across here. So, and here's our carbon dioxide being picked up by the red cells. Here we see this is a capillary wall right here, and we can see oxygen diffusing through the capillary wall there, and CO2 diffusing back out, or even coming out through these fenestrations in here at the various tissues. Because not all of the carbon dioxide is carried in our plasma. More of it is carried inside our red cells. So a couple terms here. Hypotension, low blood pressure. Not a good thing. Not a good thing if it gets too low. As long as the systolic hypotension is the systolic pressure below 100. An individual that is hypotensive has less health issues than someone who is hypertensive, but again may have issues in physical activity. Hypertension, on the other hand, has some very significant health issues here. We have several stages of hypertension where your pressure is, in the example here, 139/89. We're more concerned about the diastolic number than the systolic number, because notice how we're concerned about both. But consider, look at the stage 1. We have a systolic number of 130 to 139 and a diastolic up to 89. That doesn't sound like a very big increase, and yet that is raising more pressure than we need. You know, we're at the risk of starting to cause damage in the eyes and in the kidneys and in the peripheral tissues. Sustained blood, and we're talking about your patient comes in, blood pressure is checked, it's slightly elevated, come back in 6 months, it's still slightly, it remains slightly elevated, come back another 3 months, it's still elevated. This is a sustained elevation here. This isn't just, a fluke caused by traffic or stress or something like that. And we see a gradual increase over several years of going from 120/80 to 130/90 or higher. And we're moving into hypertension. This leads into potential damage, failure of the kidneys, a stroke in the brain, cardiovascular disease, heart disease, any number of these things, because if the pressure's up, the heart has to work harder. Heart has to work harder, the kidneys have to work harder, the pressure in the arteries in the brain is greater, and there's a chance that we can have we can have a myocardial infarction, or we can have what we call a cerebrovascular accident in the brain. So now here's something that also affects us with hypotension now. We like to keep our pressure, if your blood pressure is running like 115 over 70, that's great. Your healthcare provider is thrilled. But if we start getting consistently below 100, the problem with hypotension is that we may not be delivering nutrients and oxygen to the tissues at the rate we want. Now, here's one that we've often seen, what we call orthostatic hypotension. You get up suddenly and you're dizzy. Your blood pressure, you jump up quickly from sitting down or laying down, and your blood hasn't caught up with your brain. Your brain's suddenly here, but your blood pressure is down here. And so this is a very common problem. Older people have this easily. Our sympathetic nervous system can't keep up, doesn't respond that quickly to change in our posture. So we jump up suddenly. We're sitting in a recliner, we jump up. Well, of course, your elderly people in a recliner, that's like a 5-minute exercise to get out of the recliner. We've all seen it, right? You know what I'm talking about. The nervous system, sympathetic side, doesn't respond as quick. So we may jump up suddenly and be dizzy. Chronic hypotension is usually a result of either diet, or hypothyroidism. You're not metabolically active enough, or Addison's disease. Remember Addison's disease? The glucocorticoids, the mineral corticoids are working there against you. Now, acute hypotension can occur when your patient's bleeding out or losing a lot of blood. Yes. This may be a sign of circulatory shock. A patient who is undergoing surgery. Surgery is trauma to the body. No matter how well you describe it, surgery is an invasive procedure, and your body responds to it. It's a stress, and you may see their blood pressure dropping drastically in here. And that is an acute hypotension, and they have to be monitored. So, and I think I said a lot already. So, risk factors for hypertension: diet, obesity, age, race, heredity, stress, and smoking. Smoking can contribute to hypertension because nicotine is a vasoconstrictor. There's a lot of genetic components in hypertension. Many people, as they age, they develop hypertension because they are genetically predisposed to it. Many members of their family experience hypertension. Now, secondary hypertension is simply is caused by renin, or too much renin. That may not be their fault. It may not be something that maybe people, they've done nothing to trigger this excess renin. And so they're renin to angiotensin 1 to angiotensin 2 to aldosterone, or they may have heart disease and develop secondary hypertension. They may have an endocrine problem. They may have a tumor on the adrenal cortex causing the release of aldosterone, or causing, a tumor in the medulla causing the release of epinephrine or norepinephrine. Again, not their fault. Got to treat it, but that may not be their fault. The primary hypertension, if you take out things like race and genetics and stress, because stress can be controlled, you're left with diet, obesity, and smoking. Poor health decisions, essentially. Can't do anything about age, can't do anything about race, can't do anything about our genetics. We may be able to control our stress, but things like diet, exercise, poor choices of recreational chemicals, things like that can affect this. Okay. And okay. Circulatory shock affects our vessels too. You have a patient that's bleeding out. They have significant trauma that's affecting an artery, and they are losing a lot of blood rapidly. Unfortunately, our heart, when it sees that we're losing blood, detects that our blood pressure is dropping. So what does our heart do when our blood pressure drops? It pumps faster. So now you're pumping out blood, and the heart says, oh, well, our pressure's dropping. I better pump some more blood. And it's like making it worse. Hypovolemic shock, your blood volume is dropping drastically. That's also from your bee sting, because if the plasma is leaving your blood and going into your tissues because you're going to swell up, that fluid in the tissues was plasma a minute ago. You're reducing volume of your blood. So low blood volume. Vascular shock, extreme vasodilation. Well, again, is it the, why are we vasodilating? And in pump failure, cardiogenic shock, the heart cannot maintain circulation. We see this when the cardiac muscles have become flabby and weak and can't contract properly. And that is blood vessels. Okay. Anyone have any comments or questions on anything? Okay, then I'm going to get us out of here. As I said, lab today will be the digestive system, at least the first half of it. And we will.
Oh, I don't know if they were on when we were talking about the test.
Oh,
You had your mic on.
I think I did.
Okay.
So,